Thank you very much. Can you hear us? Sorry.
Yes. Uh-oh. You seem to have frozen.
Yeah, and microbiota has been. Sorry. Wait.
It's good now. I can hear you.
Okay, good. So should I start right now or?
Yes.
Okay, good. So yeah, the human gut microbiota has been related to many factors, but practicing sex, which is one of the most common and important human behaviors, remains barely studied. So today I will take the chance to talk more about how sexual practices could change the gut microbiota in men who have sex with men. So the initial studies suggested that HIV infections could change the human gut microbiota, but follow-up works suggested that men who have sex with men carry a microbiota which is very different from non-MSM, independently of HIV infection. And all these studies agreed on a very interesting point that MSM microbiota is enriched by Prevotellaceae family, but without a clear taxonomy resolution. Two recent metagenomic studies uncovered the understudied biodiversity in Prevotellaceae family, especially the expansion of Segatella copri complex, which was known as Prevotella copri. And this group of bacteria have been linked with many host conditions, and they are highly prevalent and abundant in non-Westernized populations. So in our study, in order to have a comprehensive insight into MSM microbiome, especially the diversity of Prevotellaceae family, we assembled a cohort of 124 subjects and recorded their anthropometrics, surveyed their sexual practices, and also deep sequenced their fecal samples. We firstly used metaflam4 to profile the taxonomy composition at the species level, especially studied the Prevotellaceae family. And then we compared MSM samples with around 1,000 samples from 10 countries. At last, we built a machine learning model to evaluate if the microbiome-based information is predictive for sexual practice. So as a result, first of all, the comparison of alpha diversity using Shannon index and the richness revealed a higher microbiome diversity in MSM compared to non-MSM. The beta diversity analysis also showed the species level microbiome composition is different between MSM and non-MSM. So we continued to check the segregation between these two groups, but stratifying the individuals into different co-variables such as BMI and age and many other factors, and we found the results turned to be consistent. Because most of our subjects are HIV patients, so we also assessed the effect of immunological parameters, including CD3, CD4, and CD8. And we found these parameters were not relevant to the microbiota change in MSM. As the high level of Prevotellaceae members has been reported as the hallmark of the MSM microbiome, we also analyzed this bacterial community. We found the average number of Prevotellaceae was 3 times higher in MSM compared to non-MSM. So was the Shannon diversity. And the beta diversity of Prevotellaceae community also showed profound segregation between these two groups. And later on, we broke down this community into individual species, and we found multiple clades of Segatella copri complex plus unknown Prevotellaceae members were enriched in MSM compared to non-MSM. But what's more interesting is the multiple clades of Segatella copri tend to occur together in MSM more frequently in non-MSM, with 70% of MSM subjects carrying at least 2 clades, in contrast to only 30% in non-MSM subjects. Similar features has been reported in non-Westernized populations as well. So these results motivated us to compare our MSM samples with other samples from other regions. So we put together around 1,000 samples from 10 countries, including 5 typical non-Westernized countries and 4 Westernized countries and urban regions in China. As a result, we found that most of our MSM samples were clustered together with non-Westernized samples. Although 97% of MSM subjects in this study have European ancestry and also have long resided in Germany, which is a typical Westernized society. Of the top 50 abundant species, MSM shared 33 species with the non-Westernized population, in contrast to only 15 species between MSM and the Westernized subjects. MSM also shared a striking similarity with the non-Westernized population in the relative abundance of Bacteroideaceae and Prevotellaceae family, and the Bacteroides and the Segatella genera, except for MSM carried even higher relative abundance of Prevotellaceae and the Segatella members. Yet there were also microbial traits likely to differentiate MSM from both Westernized and non-Westernized populations. For example, Akkermansia multifila was enriched in Westernized population compared to the men who have sex with men subjects. And Klebsiella pneumonia was enriched in non-Westernized population compared to men who have sex with men. The uniqueness of our cohort is the detailed survey for sexual practice. So in this part, we built machine learning models integrating microbiome information and sexual practice. The model evaluation revealed significant predictive power of using microbiome composition for sexual practices. It reached the highest performance in identifying the number of sexual partners. And studying the biomarkers which are related to the sexual practice of having more than 3 partners or less than 3, we found biomarkers including multiple clades of Segatella copri and other Prevotellaceae members are the main drivers. And next, we applied the same methods to other sexual practices. We found the majority of biomarkers were exclusively associated with only one specific practice. Only a few biomarkers were common to more than one practice. And after dividing all the sexual practices into the risk increasing and the risk reducing behaviors, and we found risk increasing behaviors. For example, practicing condomless sex and having more than 3 partners and others share a higher number of microbial biomarkers than risk-reducing behaviors. In order to build a link between the sexual practice and the similarity between MSM microbiome and the non-Westernized microbiome, we calculated the distance of MSM subjects to the centroids of both Westernized and non-Westernized populations. And we found sexual practices, including having more than 3 partners, practicing oral sex, and not using condom, were significant factors that promote MSM microbiota closer to non-Westernized types. So taken together, we found men who have sex with men carry a distinctive microbiota regardless of HIV infection and other anthropometric factors. And we found the Segatella copri members and other Prevotellaceae species are the main drivers. And also integrating our samples with global samples, we found MSM microbiota is highly similar to non-Westernized microbiota. And we found in this aspect, sexual practices, especially practicing oral sex and having more than 3 sexual partners and not using condom, are the significant factors. And here I just scratched the surface of the whole story. Please refer to our recently published paper for more details. And I would like to thank the contribution from our excellent colleagues, and also thank you for your time and attention.